Okay, everyone, Chapter 2 of our microbiology course is going to a little bit pick up where we left off in Chapter 1 and talk more about how we classify life and how life is organized. That's what they mean by taxonomy. And then also tools of the laboratory, just a little bit of that. We'll be going into more depth as we move on in the course. So, this chapter 1, we talked about macromolecules of life and chemistry of life and what it is to be a cell and what microbes are. Now, how do we categorize and classify life on Earth? Well, classification refers to grouping living organisms in categories that make sense. So, we want to group living things in small groups that progressively get larger in a way that we can easily understand and study them. This is what's known as taxonomy. Taxonomy is the science of doing that, identifying, naming, and classifying organisms based on phenotype, that's their outward appearance, what they look like. We might base that on biochemistry, molecules and enzymes that they have, or the metabolism that they use, and evolutionary relationships. Who's most related to who from a common ancestor? Now, taxonomy, people have been naming organisms since humans first walked the earth. But taxonomy itself really began in the 1700s with this man here. Carolus Linnaeus was a Swedish biologist that specialized in plants, and he sort of standardized taxonomy, how we name living things and the different categories that we put them in. Now, his categories have been revised several times since then, but the basic framework that Linnaeus came up with is still used today. So you can see Linnaeus just talked about plants and animals. And as time went on, we broke this down further. Today, we basically use this 3-domain system: bacteria, archaea, and eukarya. And I'll be going over that here in just a minute. So today's hierarchical system. Now, by hierarchical, we mean we can either start small and get to bigger and bigger groups, or start big and get to smaller and smaller groups down to a single species. And that's what we'll do here. The largest category that living organisms can be placed in is called a domain. There are 3 domains of living things. So all living things on Earth, including prokaryotes and microbes, will fall into one of these 3 domains. Domains archaea and bacteria contain prokaryotes. So this is where most of your microbes are, in these categories. Domain eukarya contains all eukaryotes. Again, a eukaryote is an organism made up of more advanced cells. They have a nucleus and better internal structure. Prokaryotes, simpler cells, less internal structure, and no nucleus. All eukaryotes are in domain eukarya, and some of the microbes are in here, okay, like fungi and protists and algae and things like that. Now, domains, the biggest group, are broken down into kingdoms, kingdoms into phyla, plural, phylum, singular. Phyla are broken into classes, classes into orders, orders into families, families into genera, singular is genus, and then an individual species has a specific epithet added at the end. So once we identify a species, let's say a new species, we use what's called the binomial system of nomenclature to name that organism. Now, how it works is there are 2 words that make up the scientific name of a species. The genus name and then the specific epithet. Together, it gives us the species. We usually use Latin or perhaps ancient Greek words in science, in taxonomy, to name and identify, to name organisms, because Latin is a universal language. It's not spoken conversationally anywhere in the world, so scientists across the world can use the same words and understand each other. So, for example, human beings, the scientific name for human beings is Homo sapiens. Homo is our genus. So again, if we go back here, our genus, okay? And then sapiens is our specific epithet. Together, the Homo sapiens makes up the species. Now, you will notice that the genus name is always capitalized, and the specific epithet is always lowercase, and both words are italicized. Or if you were handwriting, you could underline each word. All right, so for example, if we look here, here's a domestic cat. The whole species is Felis catus. So you see here, Felis is the genus, catus the specific epithet, together the species. And then you can see the other categories that they're in, all the way up to domain eukarya. All right, cats are eukaryotes, so they'd be in domain eukarya. All right, now the three-domain system that we use today, we put prokaryotes, as I said earlier, into two domains, domain bacteria and domain archaea, and we'll be fleshing those out in depth going forward here. Archaea, interestingly, these are small prokaryotes that resemble bacteria but are not bacteria. Archaea are more closely related to eukaryotes than they are to bacteria. All eukaryotes are in domain eukarya. And the little organelles inside of eukaryotic cells, some of the organelles that give them that internal structure I mentioned, are called mitochondria and chloroplasts, for example. Those organelles actually evolved from prokaryotes. It was a small prokaryote living inside a larger prokaryote, and that small one eventually lost the ability to live on its own and came a part of the bigger one. Fungi, interestingly, are more closely related to animals than plants. And we separate organisms into these three domains, bacteria, archaea, and eukarya, based on differences in what's called their ribosomal RNA. Now, ribosomal RNA makes up little organelles that all cells have, both eu and prokaryote, called ribosomes. All organisms have ribosomes. They're little tiny organelles where proteins are built. And the genes that make ribosomal RNA, that make up this ribosome, are not highly variable. So we can easily tell by looking at the ribosomal RNA, our RNA, whether an organism should be in domain bacteria, domain archaea, or domain eukarya. You don't have to grow cells in the lab to obtain ribosomal RNA data. You don't have to do that, so it's easier to get that data and classify an organism in those domains. Okay, so here we see a little breakdown of the three domains. Domain eukarya, this is the one we're most familiar with. We would have fungi, animals, plants, algae, and so on. Domain archaea here, which is closer to eukarya than we are to bacteria over here. And then bacteria over here. All right, now breaking this down further, what if we look more specifically at some microbial groups here? So bacterial taxonomy. Currently, there's about 3,100 species of bacteria that have been identified using conventional techniques, some things I'm going to talk about here momentarily. However, that ribosomal RNA data, that rRNA data I mentioned just before, indicates there may be millions of different prokaryotes based on genetic sequencing and gene sequencing. So there's a lot more out there than we know of right now. Now, another important point, everyone, is to understand what we mean by a strain, a bacterial strain. A strain is genetically different cells within a species. So it's the same species, but genetically different cells, like we see here. So this is Streptococcus pneumoniae colonies, smooth versus rough. So the smooth colony here, and then that rough one that looks more, more jagged on the outside, is these are two different strains of the same species, Streptococcus pneumoniae, and the same thing here. Every cell on here is E. coli, but you'll notice there are blue ones, white ones, and then the few rarer pink ones. This pink one is the strain 0157, and then the blue and the white would be different strains. All E. coli, but slightly genetically different. Archaea, there's currently about 500 known species of archaea. They're a little bit more difficult to study, so this is a small domain archaea. Archaea, as I mentioned, are more closely related to eukaryotes than they are to bacteria. And one of the reasons that archaea are difficult to study is they're often what we call extremophiles, which means they can live in very harsh environments. Very strong acid, very salty conditions, very hot, scalding temperatures, conditions that would kill most other organisms. For example, there are some archaea, example of extremophiles, called obligate halophiles that can live in extremely salty conditions, like Lake Tyrrell here in Australia. Very, very salty conditions. It's actually a pond used for a salt mine. No other living organism can really live in there except these archaea. And hyperthermophiles. Hyperthermophiles are archaea that can live in very, very hot temperatures. So you see the black smoker here. This is a hydrothermal vent at the very bottom of the ocean, where heat from Earth's core mixes with the water at the bottom of the ocean and creates these little ecosystems. Some archaea can thrive down here. Most other organisms aren't going to make it. Another group of archaea are methanogens. Methanogens are the largest group of archaea, and what they do is they can act as kind of a decomposer. They can convert organic waste in ponds, lakes, and the ocean into methane. So they break that waste down and then release methane as a gas. Some live in the colons of animals like cows and other ruminants, hooved animals that can digest cellulose. When they break down this waste, they release methane into the environment, and this is one of the primary sources of environmental methane. So if you ever see a pond like this and you're sort of just watching the pond and suddenly you see little bubbles, this little bubble, bloop, appears on the surface. That's often or likely methane gas being released from this decomposition, from these methanogens, and then it makes its way to the surface and bubbles to the surface where it's released into the atmosphere. What about viruses? Now remember, viruses are not part of the three-domain system because they're not living, okay? They're not cells. Currently about 5,000 viral species described in detail, about 220 of which are known to infect humans. Remember, they're not part of the three-domain system, so they're categorized differently. Determining taxonomy of viruses, what species they are, what groups they belong in, and so on, is very difficult because viruses can often mix RNA and DNA. They mix up nucleic acids. They mutate frequently, and they probably evolved from multiple common ancestors. So they can be tricky. There is an organization called the International Committee on Taxonomy of Viruses that classifies viruses, categorizes them, names them, and they do that by looking at what type of nucleic acid do they mostly have, DNA or RNA? Do they have an outer layer called an envelope? And what shape is their protein? Viruses are also grouped into families, genera, and they have common names. For example, okay, this is family herpes viridae, herpes virus, and the common name human herpes virus 1, 2, or 3. Another example, family Retroviridae, genus Lentavirus, and this is the genus that has HIV in it. Okay, looking like that. Here's some more families of viruses that affect humans. You don't have to know these whole figures, but just some idea on how they categorize these things. Here's a few more just for information. Okay, now the next main topic here is classification versus identification. Now, classification, as we said, is placing organisms in groups of related species, groups that make sense, that we can study. The ultimate goal, I mentioned earlier, is to categorize and classify organisms based on how closely they're related to each other from an evolutionary perspective. So we want to classify based on evolutionary relationship. Identification is matching characteristics of an unknown to a list of known. So if I have an unknown bacteria, like maybe it looks like this down here under a microscope, how do I figure out what that is? And then once I do figure out what it is, what categories does it belong in? So microbiologists use what's called the 5 I's. This is basically 5 techniques that we use to manipulate, grow, examine, characterize, and then identify an unknown microorganism. So here are the 5 I's in the order that you would use them. Inoculation. First, we're going to take a sample of our unknown and place it into a container that has some material in it on which that unknown bacteria, for example, can grow, or whatever microbe it is can grow. That's called growth media. Then we're going to incubate it. Incubation is I number 2. We're going to let this grow under proper temperature requirements, which are usually a little bit warmer, so we often use an incubator. Then we're going to isolate. So after we let it grow for a while, the species in question, the unknown, is separated from any other species that might have also been growing in that media. Then we examine that unknown species very carefully, both macroscopically and microscopically, and we do various tests to it. Finally, we can identify it based on the results of those tests. And once we identify it, we can then put it into higher categories. Now, what are the methods used to identify and classify bacteria? Now, we'll be coming back to this a little bit later, but just as a preview, one of the things we can look at are the physical characteristics. What do the bacteria look like? What's their shape? How do they group together, the arrangement? Do they take on certain dyes or stains? Do they move, etc.? We can do chemical or biochemical tests of them. Can they ferment? Can they break down sugar to make alcohol? Okay? We can use a tool called an enterotube that can do multiple biochemical tests at the same time. We can use selective media, which is growth media that favors certain microbes over others, and then differential media, which allows us to tell two or three or more different species growing on the same media. We can tell them apart. And then, of course, there's a lot others here that we can do. Here's an example of a fermentation test. This species over here, the liquid was originally red, it's still red, and there's no bubble. This in here is an upside down tube, the opening's down here. And there's no gas in here, no gas bubble. So this species does not ferment, and it does not release carbon dioxide as a gas. This one does ferment because the liquid media turned yellow, and it also releases CO2 because we've captured this bubble right here. So we would be able to tell things apart that way. These are enterotubes. Each little disc here, numbered 1 through 12, is a different type of media that will turn a different color based on what the species does. What kind of metabolism does it have? And by looking at the color patterns, we can identify an unknown. Okay? And here we see differential media. We have no bacteria growing here. This is Staphylococcus aureus, and this is Staphylococcus epidermidis, growing on the same media, but clearly we can see that the aureus has turned our medium, which was originally red, yellow. So on the same media here. We might also do a serological test. Serology, everyone, is the scientific study of serum and other bodily fluids, saliva, phlegm, things like that. And serology tends to refer to the identification of antibodies in that fluid, in that serum. An antibody is a small protein that the body produces in response to an infection. So we see here the green circles are what's called the antigen. That's what's causing the infection, say bacteria or a virus. And these purple colored things that look like letter Y's are antibodies that stick to the antigen to kind of try to destroy it. So if we detect antibodies in serum, we know that the person or the whatever organism you're studying has become infected. Otherwise, the body wouldn't make antibodies against this invader, this antigen. So we often perform a serological test when an infection is suspected, and you might also do it using what's called a slide agglutination test like this. So we put the serum on a slide, we look for antibodies. If it agglutinates or kind of clumps or clots like we see here, that's a positive test. If it doesn't, like we see here, that's a negative test. All right? If you're working clinically, you might also perform what's called a rapid strep test. This is used to see whether streptococcal bacteria are causing a sore throat. And this is your rapid strep test here. We swab the throat of the patient. Here's our sterile swab. Put it in here and look for the test results here. Okay, positive or negative. If we want to get more scientific and more specific, we can do what's called a nucleic acid analysis. This is looking at the DNA or RNA in my specimen, in my sample of interest. Now, I'm not going to get into a lot of detail here. It's beyond the scope of what you need to know and what's in this class. But suffice it to say, a wide range of techniques are used, like this is electrophoresis, where we're separating out DNA and RNA here in these individual bands. We can tag DNA with radioactive or fluorescent dye and take a look at it that way. So there's a variety of things, but this would be the most advanced technique here. This is where we're really looking right at the DNA or RNA. Now, everyone, once we collect enough data, we can put that all together using these two standard manuals in any microbiology lab. Berge's Manual of Systematic Bacteriology and Berge's Manual of Determinative Bacteriology. These would be used to characterize your unknown. Now, we're not going to find things like DNA sequences in here or anything like that, but we're going to find the results of various biochemical tests. We're going to find the results of physical examination, things like that. So you could put that all together using what's called a dichotomous key, something like this, where we start by doing a gram stain or gram reaction. Is that positive or negative? Depending on that basic result, we go this way or this way, and then do a variety of tests, okay, and work our way down this key until we get an answer. I would then check that answer in these manuals. All right. Viruses, identifying them can be a little trickier. We often just look at signs and symptoms and whether or not antibodies are present. Like, for example, with shingles or other herpes infections, we're looking for antibodies. Sometimes we have to do that nucleic acid analysis, DNA or RNA, like for West Nile virus. So again, bacteria are a little bit easier. There's more tests to identify them than it is for viruses. Okay? All right, everybody, that will do it for Chapter 2 here, Taxonomy and some basic tools of the laboratory. In our next chapter, we're going to focus very specifically on prokaryotes and bacteria. So that is it for today, for this chapter. And until next time, you have a great rest of the day. Bye-bye.